Honey is one of those quiet, familiar foods that seems simple at first glance, yet carries a long and slow history through nature and human life. If you think about it gently, honey is not really something that begins in a factory or even a kitchen. It begins in fields of flowers, in warm air, and in the small, repetitive movements of bees traveling between blossoms. Each drop is the result of countless tiny journeys, where nectar is gathered in small amounts and slowly transformed over time inside the hive. For something so sweet and smooth, its creation is surprisingly patient. Nothing about it is rushed. Even the way it settles into a jar feels steady, thick and unhurried, as if it remembers the quiet rhythm of the hive. When we look at honey from a scientific perspective, it becomes less like a simple sweetener and more like a natural record of plant life, weather patterns, and the gentle coordination of insects working together in a way that has remained largely unchanged for millions of years. Inside honey, most of what we experience is a carefully balanced mixture of natural sugars, mainly glucose and fructose. These simple sugars are already broken down compared to more complex carbohydrates, which is part of why honey tastes immediately sweet and feels smooth on the tongue. But beyond sweetness, honey also contains small traces of enzymes introduced by bees, along with tiny amounts of minerals, pollen particles, and organic compounds that vary depending on the flowers visited. This variation is one reason honey from different regions can taste slightly different even when the overall structure remains similar. From a physical point of view, honey is also highly viscous, meaning it resists flow and moves slowly under gravity. This thickness comes from its low water content and dense sugar concentration. In a quiet way, it behaves almost like a slow-moving liquid memory of the landscape it came from, holding within it both botanical detail and biological transformation. When honey is consumed in small amounts, the body processes it in a relatively straightforward way. The simple sugars can be absorbed without the long breakdown process required by more complex starches, which means energy becomes available in a gradual but noticeable manner. However, honey is still a sugar-rich substance, and its effect on the body depends greatly on quantity and context. In calm nutritional terms, it is often described as having a moderate glycemic impact compared to refined sugar, partly due to its composition and trace compounds. Some people find that a small amount in warm water or tea feels steady rather than sharp in its energy effect, though responses can vary widely from person to person. From a physiological standpoint, the body is always working to maintain balance in blood sugar levels, and honey simply becomes one of many natural inputs that the system processes, adjusts to, and integrates into its ongoing rhythm of metabolism without needing anything dramatic or sudden. If we step back and think about honey more slowly, beyond its chemistry and biology, it becomes a quiet example of transformation in nature that does not rely on force or urgency. Nectar is gathered drop by drop, carried carefully, then gradually refined inside a dark, structured space where bees regulate temperature, airflow, and moisture with consistent precision. Over time, something watery and light becomes dense and stable, capable of lasting long periods without spoiling easily. This slow concentration process is not just chemical, but environmental and behavioral, shaped by the coordinated actions of an entire colony. Even the way honey rests in stillness reflects that history, as if every layer of it remembers the gradual evaporation of water and the repeated movements of wings inside the hive. Observing it closely can feel like watching time become stored in a physical form, not in a symbolic way, but in a very literal biological sense, where process and result are gently compressed into one substance. And in that idea alone, there is something quietly settling. Honey does not ask for attention, and it does not change quickly once formed. It simply remains, stable and consistent, shaped by repetition and patience at a scale that is almost invisible. When it is seen under soft light, it often appears still, almost suspended, as if it prefers to exist in a slower register of time. Thinking about it in this way can make the mind itself feel slightly less hurried, as if the structure of nature includes places where change is gradual enough to almost pause. In the end, honey becomes less about sweetness alone and more about continuity, a small reminder that some of the most persistent forms in nature are built not from intensity, but from quiet accumulation over long, uninterrupted stretches of time. If we continue staying with honey a little longer, there is another quiet layer of science that becomes visible, one that has to do with how stable it is over time. Unlike many natural liquids, honey resists spoilage in a way that feels almost unusual at first, yet it follows simple physical principles. One of the main reasons is something called low water activity. Even though honey looks fluid, it actually contains very little free water available for microbes to use. Most of the water is tightly bound within the dense sugar structure, especially the high concentration of glucose and fructose. This creates an environment where bacteria and many microorganisms struggle to grow or reproduce. In a gentle sense, honey becomes less like a welcoming habitat and more like a tightly controlled landscape where biological activity is naturally slowed down. There is also a mild production of hydrogen peroxide when honey is diluted, created through enzymes introduced by bees, which adds another layer of protection. It is not a harsh or aggressive defense, but rather a subtle chemical background process that contributes to its long shelf stability. All of this happens without any artificial intervention, shaped entirely by natural chemistry and the behavior of bees within the hive. Inside the hive itself, honey exists in an environment that is carefully maintained without conscious planning, yet remarkably consistent. Bees regulate temperature with surprising precision, often keeping the hive warm enough for development, but stable enough to protect stored food. They fan their wings to circulate air, adjust moisture levels by evaporating excess water from nectar, and seal honey into wax cells once it reaches the right consistency. This sealing process is especially important because it protects honey from absorbing too much moisture from the air, which would otherwise change its stability. Over time, each sealed cell becomes a small storage unit of concentrated energy, preserved in a state that can last for months or even years. When we think about it slowly, the hive is not just a home, but also a living preservation system where biological activity and environmental control are intertwined. Honey is not separate from this system, it is a direct expression of it, shaped by countless small adjustments made by thousands of bees working in parallel, each contributing to a result that feels smooth and unified when we eventually see it outside the hive. There is also a quiet physical change that honey undergoes depending on temperature and time, known as crystallization. This process can seem surprising if it is not expected, but it is entirely natural. Over time, especially in cooler environments, the glucose in honey can begin to form small crystals, while the fructose remains more fluid. This causes the texture to gradually shift from smooth and flowing to thicker and more granular. It is not a sign of spoilage, but rather a rearrangement of structure at the molecular level. If gently warmed, these crystals can dissolve again, returning honey to a more liquid state. In a slow scientific sense, this shift reflects how even stable substances continue to move internally, responding to temperature and time in subtle ways. Nothing in honey is truly static. Even when it appears still, there is always a quiet internal motion of molecules, always a gentle balance between order and change. And yet, this transformation never feels abrupt. It unfolds gradually, almost imperceptibly, as if the substance itself prefers to avoid sudden transitions. When honey enters the human body, its journey continues in a similarly calm and steady way. The simple sugars it contains are absorbed through the digestive system and enter the bloodstream, where they become part of the body's ongoing energy regulation. Fructose and glucose follow slightly different pathways in metabolism, but both are ultimately used as fuel for cellular processes. What is interesting is not any single dramatic effect, but the way honey integrates into a larger biological rhythm that is already ongoing. The body is constantly balancing energy intake and usage, and honey simply becomes one of many inputs that fit into this continuous process. Some of its trace compounds, such as small amounts of pollen or plant-derived antioxidants, interact subtly with the digestive environment, though their effects are generally mild and dependent on quantity. From a scientific perspective, What stands out is not intensity but compatibility. Honey does not disrupt the system. It enters it and is gradually incorporated, becoming part of the same quiet metabolic flow that sustains life moment by moment. If we stay with honey a little longer, we begin to notice how closely its existence is tied to seasons, even when it sits quietly in a jar far away from any field or flower. The composition of honey is never completely fixed because it depends on what plants were flowering at the time it was collected. In spring, when blossoms are abundant and diverse, honey tends to carry a lighter and more varied floral profile. In late summer, when certain plants dominate the landscape, the same bees may produce honey with a deeper, more concentrated character. This seasonal imprint is not something added later, but something captured in real time as nectar is gathered. In a very slow scientific sense, honey becomes a kind of natural archive of the environment, preserving chemical traces of plant life that existed during a specific window of time. Even pollen grains, sometimes present in microscopic amounts, can reflect the botanical history of the region. When we think about this gently, it is almost as if honey is not only food, but also a quiet record of ecological conditions, holding within it subtle information about weather, geography, and the cycles of plant growth that shaped its origin. There is also something interesting about the way honey interacts with light and temperature in its physical form. When light passes through honey, especially in a calm environment, it slows slightly due to its density and refractive properties, creating a soft golden appearance that feels visually steady rather than sharp. This is not just aesthetic, it comes from the way sugar molecules are packed closely together, bending and scattering light in a consistent pattern. Temperature also plays a large role in how honey behaves. At warmer temperatures, the molecules gain slightly more movement, allowing honey to flow more easily. At cooler temperatures, this motion slows and the substance thickens. These shifts are gradual and predictable, governed by basic principles of thermodynamics. Nothing in honey reacts suddenly unless the change in environment is sudden. Otherwise, it responds gently, adjusting its viscosity as if it is always trying to remain in balance with its surroundings. In this way, honey can be thought of as a substance that reflects its environment rather than resisting it, subtly mirroring the conditions around it through small physical changes that unfold over time. If we move even deeper into its biological origin, honey also reveals something about the communication and coordination within a bee colony. Bees do not produce honey individually in isolation. Instead, it is the result of a shared process involving foraging bees, worker bees, and the internal structure of the hive. Foraging bees collect nectar and pass it along through a chain of transfer, where it is repeatedly handled, partially digested, and enriched with enzymes. This cooperative process gradually transforms nectar into a more stable substance. The famous waggle dance is one of the ways bees communicate the location of flowers, allowing the colony to efficiently gather resources without central planning. Each bee responds to local signals, yet the overall outcome is highly organized. From a systems perspective, honey is not just a product of biology but also of distributed intelligence, where simple rules followed by many individuals result in a highly structured and functional outcome. This type of organization appears in many natural systems, but in the case of honey, it becomes physically stored and visible, making the process easier to observe in its final form. As honey sits outside the hive, its stability continues to reflect this long chain of biological cooperation. Even in storage, it remains chemically resistant to many forms of decay, not because it is inert, but because its internal structure is carefully balanced. The high sugar concentration creates osmotic pressure, which draws water out of microbial cells, making it difficult for them to survive. This effect, combined with low moisture and mild acidity, contributes to its long-lasting nature. Yet despite this stability, honey is not truly frozen in time. It can slowly absorb moisture from the air if left uncovered, gradually shifting its properties. It can also darken slightly over long periods due to natural chemical reactions between sugars and amino acids, known as the mild reaction, which occurs slowly at room temperature. These changes are subtle and often take place over months or years, reinforcing the idea that even stable natural substances continue to evolve quietly when given enough time. And so, when we observe honey in its stillness, whether in a hive, a jar, or a spoon held in soft light, we are not looking at something static. We are looking at a condensed outcome of movement, interaction, and time. Every part of its existence is shaped by slow processes that rarely demand attention in the moment they occur. Flowers release nectar gradually. Bees collect it in repeated cycles. Enzymes transform it step by step, and environmental conditions shape its final form without urgency. Even now, in its resting state, honey continues to respond quietly to temperature and air, maintaining a delicate balance that has been refined over countless cycles of natural repetition. In a calm scientific sense, it reminds us that some of the most stable things in nature are not the result of stillness itself, but of long, continuous motion that has simply settled into a form we can hold in a single moment. When we look more closely at honey's long stability, one of the quiet scientific layers beneath it is its gentle antimicrobial behavior. This does not come from a single strong ingredient, but from several mild properties working together in balance. The high sugar concentration creates what scientists call osmotic pressure, which draws water away from microorganisms and makes it difficult for them to maintain their structure. At the same time, honey is naturally slightly acidic, which further limits the growth of many bacteria. In addition, when honey is diluted, an enzyme called glucose oxidase can slowly produce small amounts of hydrogen peroxide, creating a mild chemical environment that discourages microbial activity. None of these effects are extreme on their own, and they do not make honey sterile in a strict sense, but together they create a setting where microbial life is naturally restricted. This is one reason honey has been used in some traditional and modern wound care contexts under controlled medical conditions, where its properties can help maintain a clean environment. From a scientific perspective, what is important is not any single dramatic effect, but the layered combination of gentle mechanisms that reinforce each other over time, forming a natural system of preservation without requiring intensity. If we shift gently from chemistry to perception, honey also interacts with the human senses in a slow and layered way. Its taste is not experienced all at once, but unfolds gradually across the tongue as different sugar molecules activate sweet receptors with slightly different timing. The aroma of honey also carries subtle volatile compounds that come from the original flowers, and these are detected by olfactory receptors that send signals to the brain's sensory regions. This combination of taste and smell creates what we perceive as flavor, but even that experience is not static. It changes slightly as honey warms in the mouth, dissolves, and mixes with saliva. The brain does not just register sweetness, it integrates memory, expectation, and sensory input into a single perception that feels smooth and continuous. In a calm scientific sense, eating honey becomes a slow sensory event rather than a sharp one. Each moment of perception blends into the next, with no clear edges, as if the experience itself prefers to remain soft and extended rather than sudden or fragmented. This is part of why honey often feels gentle on awareness, not because it is absent of intensity, but because its signals arrive in a steady, layered rhythm. Behind all of this sensory experience is the quiet intelligence of the bees that created it, operating through a system that does not rely on central control. Inside a hive, thousands of individual bees respond to local cues: temperature, scent, vibration, and movement. When a forager finds a rich source of nectar, it returns and communicates direction and distance through a patterned movement known as the waggle dance. This dance is not symbolic in a human sense, but functional, encoding information that other bees interpret and act upon. What emerges from this is a distributed system where no single bee directs the entire process, yet the colony as a whole behaves with remarkable coordination. Nectar collection, transformation, and storage happen through repeated interactions between individuals, each contributing small steps that accumulate into a stable outcome. Honey, in this sense, is not just a substance produced by bees, but a physical record of collective behavior. It reflects a system where organization arises from repetition and local responsiveness rather than centralized instruction. When viewed slowly, it becomes an example of how complex order can emerge from many simple actions occurring in parallel over time. This collective process is deeply connected to the broader environment because bees themselves are embedded within ecosystems that depend on plant diversity and seasonal cycles. As bees move between flowers, they participate in pollination, transferring pollen grains that allow plants to reproduce. This interaction supports not only the production of nectar, but also the continuation of flowering species that shape entire landscapes. In this way, honey indirectly reflects the health and variety of the environment from which it comes. Changes in available flowers, climate conditions, and land use can all influence the character and quantity of honey produced in a region. From a scientific perspective, honey can sometimes serve as a subtle indicator of environmental conditions, since its composition is linked to the plants that were available during its formation. It is not a precise measurement tool, but it carries embedded traces of ecological activity. When considered slowly, honey becomes part of a larger cycle where plants, insects, and climate are interconnected, each influencing the others in continuous feedback over time. And so, as we stay with this idea a little longer, honey begins to feel less like a finished object and more like a pause in a much larger process. It is a moment where movement has been gathered and gently held in place, without being fully separated from its origins. Even now, it continues to respond softly to its surroundings, while still preserving the long sequence of events that brought it into existence. In a quiet way, it reminds us that natural systems rarely operate in isolation or finality. Instead, they unfold through cycles that overlap, extend, and repeat, forming patterns that are only briefly visible when they settle into something as simple and familiar as honey resting in still air. When we continue to observe honey from a calm scientific distance, another subtle aspect begins to appear in the way it stores and releases energy. Honey is often described as a concentrated energy source for bees, and this is not just a poetic idea, but a direct reflection of its chemical structure. The sugars in honey, primarily glucose and fructose, contain chemical bonds that store energy from the original sunlight captured by plants through photosynthesis. In that sense, honey is indirectly a stored form of solar energy, transformed first by plants into nectar and then refined by bees into a denser, more stable material. When bees consume honey, or when other organisms metabolize it, those bonds are gradually broken down, releasing energy that can be used for movement, maintenance, and survival. This entire process is part of a continuous energy flow that begins with sunlight, passes through plants, enters insects, and eventually becomes part of broader ecological cycles. Nothing in this system is isolated. Honey is simply one of the more stable intermediate forms in a long chain of energy transformation that quietly connects the sun to living activity on Earth. If we slow the perspective further, even the texture of honey begins to reveal something about molecular behavior. Its thickness, or viscosity, is not fixed in an absolute sense. Instead, it is the result of countless sugar molecules interacting with each other and with the small amount of water present. These interactions create internal resistance to flow, meaning that when honey moves, it does so slowly because each layer must shift past another layer with gentle friction. This is why honey pours in a continuous ribbon-like stream rather than splashing or separating quickly like water. At a molecular level, nothing is rigid or solid, yet the collective effect produces a sense of heaviness and steadiness. Temperature influences this behavior significantly. As warmth increases, molecular motion becomes more active, reducing resistance and allowing honey to flow more freely. As it cools, molecular movement slows, and the structure becomes more resistant to change. This simple relationship between temperature and motion reflects a broader principle in physics, where the behavior of matter is always shaped by the balance between energy and structure. Honey simply makes this balance easy to observe in everyday life. There is also a quiet temporal aspect to honey that becomes more noticeable when we consider how slowly it changes under normal conditions. Unlike many foods that quickly break down or lose their structure, Honey can remain relatively stable for very long periods when stored properly. Archaeological findings have even discovered honey in ancient tombs that still retained its basic chemical integrity after thousands of years. While not necessarily suitable for modern consumption in every case, these findings demonstrate the extraordinary stability of its composition. This stability does not come from inactivity, but from a kind of chemical equilibrium where most of the components are already in a low reactivity state. Water activity is minimal, microbial growth is restricted, and the sugars themselves are relatively stable under normal temperatures. Over time, slow processes like crystallization or mild flavor darkening may occur, but these changes unfold gradually rather than abruptly. In this way, honey behaves like a substance that resists rapid transformation, preferring instead to shift in small, almost imperceptible steps across long stretches of time. If we bring attention back to the natural world where honey originates, the role of flowers becomes more central again. Nectar itself is not a byproduct, but a deliberate biological offering from plants, designed to attract pollinators. It contains sugars and aromatic compounds that signal availability and guide insects toward it. When bees collect nectar, they are participating in a mutual exchange that benefits both species. Plants gain assistance in reproduction through pollination, while bees gain access to a reliable energy source. This relationship is not based on intention in a conscious sense, but on evolutionary adaptation over long periods of time. Over millions of years, plants that produced nectar attracted more pollinators, increasing their chances of reproduction, while bees that efficiently collected and stored nectar had better survival rates. Honey, then, is not just a substance created by bees alone, but a result of long-term co-evolution between insects and flowering plants. It is a shared outcome of two biological systems adapting to each other in a continuous cycle of interaction and refinement. And when all of this is considered together, honey begins to feel like a quiet convergence point, where many natural processes meet. Sunlight becomes plant energy becomes nectar, nectar becomes honey, and honey becomes stored potential that can later return to motion through consumption and metabolism. Each step in this sequence is gradual, with no sharp boundaries between stages. Even now, when honey rests in stillness, it is not disconnected from this chain. It remains a preserved moment within a larger flow, holding within it the memory of transformation without actively changing at a visible pace. In a calm scientific sense, honey reminds us that stability in nature is often not the absence of change, but the result of changes so slow and balanced that they appear still to the human eye. If we gently follow honey into human history, it begins to appear not as a modern pantry item, but as one of the earliest natural sweet substances ever widely encountered. Long before refined sugar existed, honey would have been one of the most direct ways early humans experienced concentrated sweetness in nature. Its appearance in ancient cave paintings and early written records suggests it held a consistent presence across many early cultures, not only as food but as something observed with quiet respect. The reason is not difficult to understand when seen through a simple scientific lens. In a world without refrigeration or industrial preservation, a substance that naturally resists spoilage would have stood out immediately. Honey's stability, combined with its energy density, made it uniquely valuable. Yet what is more interesting is not just its usefulness, but how little it needed to be altered. Unlike many other natural resources that required complex processing, honey could be gathered and used almost exactly as it was found, already transformed by bees into a form that was stable, portable, and ready for consumption. In this way, it quietly bridges the natural world and early human survival without needing further refinement. When we consider honey in terms of human perception, there is also a subtle neurological aspect to how it is experienced. The sensation of sweetness is detected by specialized receptors on the tongue that respond primarily to sugar molecules. These signals travel through neural pathways to the brain, where they are interpreted as taste. But this process is not purely mechanical. The brain integrates taste with smell, texture, temperature, and even expectation, forming a unified sensory impression. Honey, because of its viscosity and aromatic complexity, engages multiple sensory channels at once in a slow, layered manner. Unlike sharp or fragmented tastes, it tends to unfold gradually in perception, as it spreads across the tongue and warms slightly in the mouth. This extended interaction gives the brain more time to process each component of the experience, blending them into a continuous sensory flow. From a neurological perspective, this kind of input is not about intensity but about integration, where multiple small signals merge into a single smooth perception that changes gently over time rather than abruptly. If we move deeper into the microscopic structure again, honey also contains a quiet chemical diversity that is often overlooked. Beyond glucose and fructose, there are trace compounds such as organic acids, amino acids, and plant-derived polyphenols. These are present in very small amounts, yet they contribute subtly to honey's overall chemical character. The exact composition varies depending on the floral sources, meaning no two batches are perfectly identical. This variation is not random but reflects the environmental conditions at the time of production. Rainfall, soil composition, plant species, and even temperature can influence the final profile. From a scientific point of view, honey becomes a kind of composite signal of its environment, carrying within it small chemical signatures of the landscape it originated from. Even though these signals are not easily interpreted in everyday life, they remain embedded within its structure. In a slow observational sense, honey can be seen as a condensed environmental snapshot, where biological and chemical processes from a specific place and time are preserved together in a single, stable form. There is also another quiet transformation that honey undergoes over long periods, especially when stored in varying conditions, and that is the gradual shift in its internal structure due to crystallization dynamics. This process is influenced by the ratio of glucose to fructose, as well as temperature and the presence of microscopic particles that can act as nucleation points. Over time, glucose molecules may begin to organize into small crystalline structures which spread slowly through the substance. This does not change its fundamental nature, but it does alter its texture and flow. What is interesting is that this process is not uniform. It can begin in isolated regions and expand gradually, creating a slow reorganization of structure at a microscopic level. Even then, honey remains chemically stable, and the process can often be reversed with gentle warming. In a broader sense, this reflects how natural substances can exist in multiple states of internal order without losing their identity, shifting between smooth and granular forms while remaining fundamentally the same material. And when all of these layers are held together, honey appears less like a simple food and more like a long, continuous intersection of biology, chemistry, ecology, and time. It carries within it the memory of flowers, the coordination of insects, the stability of molecular structures, and the slow influence of environmental change, even in stillness. It remains connected to all of these processes, not as a symbol, but as a physical continuation of them. In a quiet scientific reflection, honey can be seen as a reminder that many of the most stable and familiar things in nature are not separate from motion, but are instead the result of motion that has been gently accumulated, balanced, and preserved until it becomes something we can hold in a single moment of calm observation. If we continue to stay with honey, there is still another layer of quiet science that appears when we think about how it behaves under long periods of rest. Even when it seems completely still inside a jar, honey is not truly motionless. At the molecular level, there is always a very slow rearrangement of particles, a subtle internal adjustment driven by temperature fluctuations in the surrounding environment. These changes are not dramatic enough to alter its overall appearance quickly, but they are part of the reason honey can slowly shift in texture or clarity over time. In warmer conditions, molecules gain slightly more kinetic energy, which allows the substance to remain more fluid. In cooler conditions, that motion decreases, and the internal structure becomes more resistant to flow. This continuous but gentle responsiveness to temperature gives honey a kind of quiet adaptability. It does not resist its environment forcefully, nor does it change abruptly. Instead, it adjusts in small increments, maintaining a stable identity while still responding to external conditions in a soft and gradual way. If we look more closely at the way honey interacts with water, another important scientific property appears. Because of its high sugar concentration, honey naturally attracts and absorbs moisture from the surrounding air when exposed. This is due to a process known as hygroscopy, where substances draw water molecules toward themselves because of chemical affinity. In a sealed environment, this effect is minimal, but when honey is left open, it can slowly change its moisture balance over time. This is one of the reasons storage conditions matter so much for maintaining its stability. Too much moisture can alter its internal balance and potentially allow fermentation processes to begin, where natural yeasts present in the environment may slowly convert sugars into alcohol and carbon dioxide. However, under normal dry conditions, this does not typically occur. Instead, honey remains in its stable, concentrated form, continuing its long-term preservation without significant change. This delicate relationship with moisture is another example of how honey exists within a narrow range of equilibrium, where small environmental shifts can gently influence its behavior without disrupting its overall structure. There is also something quietly interesting in how honey reflects light when it is observed in different contexts. Its golden appearance is not just a simple color, but the result of how light interacts with a dense, translucent medium. As light enters honey, it slows down compared to its speed in air, bending slightly as it passes through the thick sugar solution. This bending, combined with internal scattering caused by tiny particles and variations in density, produces the soft amber glow often associated with honey. The exact shade can change depending on its floral origin and age, ranging from pale gold to deep amber tones. Over time, slight chemical changes can darken its color subtly, especially if it is exposed to heat or light for extended periods. These changes occur gradually and are part of the natural aging process of organic substances. From a physical perspective, honey becomes a medium where light is gently filtered and softened, rather than sharply reflected or absorbed. This contributes to its calm visual quality, where even its appearance seems to avoid harshness or sudden contrast. If we return again to its biological origin, the efficiency of honey production inside a hive becomes even more remarkable when viewed as a distributed system. Bees do not store nectar in its final form immediately. Instead, it passes through multiple stages of transformation. First, it is collected from flowers and stored temporarily in a specialized stomach, where enzymes begin breaking down complex sugars into simpler forms. Then it is transferred to other bees within the hive, who continue the process of enzymatic modification and gradual dehydration. Finally, it is deposited into wax cells, where airflow created by wing movement helps evaporate excess water. Each of these stages is essential, and none can produce honey alone. The transformation is gradual and cooperative, relying on repeated interactions between individuals and the physical environment of the hive. What emerges is not just a stored resource, but a refined substance shaped by layered biological activity. In this sense, honey can be understood as a process that has been slowed down and stabilized into a final form, preserving the sequence of its own creation within its structure. And when all of these perspectives are gently combined, honey begins to feel like a quiet intersection where multiple natural principles meet and remain in balance. Chemistry provides stability, biology provides transformation, physics explains flow and light interaction, and ecology provides context through plant and insect relationships. None of these layers dominate the others. Instead, they coexist in a steady arrangement that allows honey to remain both stable and responsive at the same time. Even in stillness, it carries the memory of movement, and even in simplicity, it holds a long chain of interconnected processes. In a calm scientific reflection, honey becomes a reminder that many of nature's most familiar substances are not defined by a single property, but by the gentle harmony of many processes unfolding together over time. If we continue following honey through its quiet scientific story, another layer appears in the way it responds to time, on a scale that is longer than a single season. Over months and years, Honey can slowly undergo subtle changes that are not immediately visible, but still part of its natural progression. One of these is the gradual development of darker color tones. This occurs mainly due to slow chemical reactions between sugars and trace amino acids, often referred to as non-enzymatic browning reactions. These reactions do not require any living organisms, but instead depend on time, temperature, and the internal chemistry of the substance itself. As they progress at a very slow rate, they can gently shift honey's appearance from lighter golds toward deeper amber shades. This change is not a sign of deterioration in the simplest sense, but rather a reflection of ongoing chemical interactions that continue even in a stable system. In a calm, scientific way, it shows that even when a substance appears settled, it can still carry forward quiet internal processes that unfold gradually over extended periods. There is also the aspect of aroma that subtly evolves with time. Honey contains many volatile compounds that originate from the flowers visited by bees. These compounds are responsible for the delicate floral notes that can sometimes be detected when honey is fresh. Over time, some of these volatile elements can slowly dissipate or transform, leading to slight changes in aroma profile. This does not happen suddenly, but through slow diffusion and natural chemical rearrangement. Temperature plays an important role in this process as well, since warmer conditions can accelerate the movement and evaporation of aromatic molecules. In cooler and stable environments, these changes are much slower, allowing honey to retain its original scent for longer periods. From a sensory science perspective, aroma is always a dynamic interaction between material composition and environmental exposure. Honey simply demonstrates this in a more gradual and extended way, where even its scent evolves gently rather than shifting abruptly. If we move back into the microscopic structure again, honey's relationship with crystallization becomes even more detailed. The formation of crystals is not random, but follows predictable patterns based on molecular arrangement. Glucose molecules tend to organize into structured lattices when conditions allow, especially when temperature drops or when water content is relatively low. These crystals can begin as tiny nucleation points, often around microscopic particles or air bubbles suspended in the honey. From these small origins, crystallization can slowly spread outward, creating regions of semi-solid texture within the otherwise fluid matrix. However, this process is reversible in many cases. Gentle warming can provide enough energy to disrupt the crystal structures, allowing them to dissolve back into the liquid form. This reversible nature highlights the flexibility within honey's internal organization. It is not locked into a single state, but exists in a dynamic balance between order and fluidity, where both structures can coexist depending on environmental conditions. There is also a quiet physical principle that becomes visible when honey is poured in slow motion. The way it stretches into long, continuous threads is a result of cohesive forces between molecules being stronger than the force of gravity pulling it downward at that moment. This creates a temporary resistance that allows honey to elongate before breaking. The point at which it finally separates is determined by the balance between internal cohesion and external force. This is why honey often forms thin, tapering strands rather than breaking cleanly like less viscous liquids. In fluid dynamics, this behavior is an example of how viscosity influences flow patterns, but in simple observation, it appears as a slow stretching motion that gradually narrows until it detaches. Even in this small action, honey demonstrates how physical laws operate continuously at different scales, shaping behavior in ways that are predictable but still visually gentle. If we widen the perspective once more, honey's existence continues to reflect the relationship between stability and change in natural systems. It is stable enough to persist over long periods, yet flexible enough to respond to environmental shifts. It is produced through biological activity, yet preserved through physical and chemical constraints. It exists as both a product and a process, depending on how closely one looks at it. This dual nature is not unusual in nature, but honey makes it especially visible because of how familiar and accessible it is. It sits quietly in everyday life, yet contains within it layers of transformation that span biology, chemistry, physics, and ecology all at once. In this sense, honey becomes less of an isolated substance and more of a convergence point, where multiple natural processes settle into a form that remains stable enough to be observed, studied, and experienced without urgency. And so, as we continue to reflect on it, honey remains in a kind of calm equilibrium within this broader understanding. It does not demand attention, nor does it change quickly enough to disrupt perception. Instead, it holds its place gently within the natural world, carrying within it the accumulated effects of many slow processes that extend far beyond a single moment. In a quiet scientific sense, it reminds us that some of the most enduring forms in nature are not defined by stillness alone, but by the long, patient alignment of many small changes that eventually settle into something stable enough to remain. If we continue to stay with honey and allow its science to unfold even more slowly, we begin to notice how its behavior also reflects something about long-term equilibrium in natural systems. Honey does not exist in a perfectly frozen state, but rather in a condition where many opposing tendencies are balanced. Sugar molecules naturally attract water, yet the overall structure remains dry enough to prevent easy microbial growth. Temperature encourages movement, yet viscosity resists it. Environmental moisture tries to enter, yet internal concentration pushes water activity outward. These opposing influences do not cancel each other in a dramatic way, but instead settle into a stable middle range. In scientific terms, this is a form of dynamic equilibrium, where processes continue to occur, but at rates that keep the overall system steady. What makes honey interesting in this context is that its stability is not dependent on being inactive, but on the continuous balancing of subtle forces. It remains stable not because nothing is happening inside it, but because everything happening inside it is gently balanced against everything else. If we look even closer at the internal chemistry, honey also contains small amounts of acids such as gluconic acid, which contribute to its mild acidity. This acidity plays a quiet but important role in its overall stability. Many microorganisms prefer neutral environments, and even slight acidity can make conditions less favorable for their growth. The production of gluconic acid itself is connected to enzyme activity introduced by bees, particularly glucose oxidase, which slowly transforms glucose under specific conditions. This is not an aggressive or rapid reaction, but a gradual process that unfolds over time, especially when honey is diluted or exposed to moisture. In its concentrated form, however, these reactions remain limited, contributing to honey's long-lasting nature. From a chemical perspective, honey can be seen as a carefully tuned environment, where pH, sugar concentration, and enzymatic residues all interact in a way that supports long-term preservation without requiring external intervention or artificial control. There is also a subtle aspect of diffusion that takes place within honey, even when it appears completely still. Molecules within any liquid are always in motion at a microscopic level, moving randomly and slowly spreading out over time. In honey, this movement is significantly slowed due to its viscosity, but it does not stop entirely. Over long periods, this can lead to very gradual redistribution of components within the substance. For example, differences in temperature or concentration can slowly even out as molecules shift through internal space. This is not something that can be easily observed in short time frames, but it is part of the underlying physical reality of all fluids. In honey, this process is simply extended into a much slower timescale. It is as if motion has not been removed, but stretched thin across time, becoming nearly imperceptible while still present in the background. If we return once again to the environment where honey originates, the role of seasonal variation becomes even more significant when considered over multiple years. Bees do not collect nectar from a fixed source. Instead, they respond to shifting floral availability across landscapes that change with temperature, rainfall, and ecological conditions. As a result, honey produced in different years, even in the same location, can show variation in flavor, color, and composition. This variation is not random, but reflects the ecological state of the environment at the time of production. In this sense, honey becomes a kind of repeating but never identical record of seasonal cycles. Each batch carries within it subtle differences that correspond to the conditions under which it was formed. Over long periods, this creates a layered continuity where each jar of honey is both similar to and distinct from others, connected through process but differentiated through time. If we expand this idea further, honey can also be seen as part of a broader pattern of energy storage in ecosystems. Plants convert sunlight into chemical energy through photosynthesis. Nectar is one of the ways this energy is made accessible to other organisms. Bees then refine and concentrate this energy into honey, creating a more stable and long-lasting form. This stored energy can later be used by bees during periods when foraging is not possible, such as colder seasons or times of scarcity. In this way, honey functions as a buffer within ecological systems, smoothing out fluctuations in resource availability. It does not create energy, but it redistributes and concentrates it in a form that can be accessed later. From a systems perspective, this represents a natural method of energy management, where biological processes align with environmental cycles to maintain continuity across changing conditions. And when all of these observations are held together gently, honey appears as a substance defined not by a single property, but by a long alignment of interactions that span multiple levels of nature. It is shaped by plant biology, refined by insect behavior, stabilized by chemistry, and influenced by physics at every stage of its existence. Even in stillness, it remains connected to these layers, not as separate parts, but as a continuous structure that has simply slowed down enough to be observed. In a quiet scientific reflection, honey becomes a reminder that stability in nature is often not a fixed condition, but a long, ongoing balance of forces that continue to exist even when the system appears completely at rest. If we continue to follow honey through an even slower scientific lens, one of the most subtle ideas that begins to emerge is how it behaves as a form of long-term biological memory. Not memory in a conscious sense, of course, but in the way it preserves traces of the processes that created it. Every stage of its formation leaves behind small chemical signatures, and these signatures remain embedded in its structure long after the activity itself has ended. The floral sources contribute distinct combinations of sugars and aromatic compounds. Bees contribute enzymes and structural transformations, and the environment contributes temperature and humidity conditions that influence how quickly water is removed. All of these influences are captured and retained in a stable form. In this way, honey can be understood as a condensed record of multiple interacting systems, where each layer of its composition reflects a different stage of its formation. Even though it appears uniform at a glance, it carries within it a layered history that can be studied through careful analysis of its chemical profile. If we move gently toward its physical interaction with time again, honey also demonstrates an interesting resistance to abrupt change. Many substances respond quickly to environmental shifts, but honey tends to adjust gradually. This is largely due to its high viscosity and dense molecular structure, which slow down internal movement. When temperature changes, the response is not immediate throughout the entire substance. Instead, heat or cold spreads gradually from the outside inward, creating slow gradients of motion. This means that different parts of a single container of honey may briefly exist in slightly different physical states, with variations in flow and texture that eventually even out over time. This slow internal adjustment reflects a broader principle in thermodynamics, where systems tend to move toward equilibrium, but the rate at which they do so depends on their internal structure. In honey, that structure naturally favors slow transitions, giving it a calm, almost deliberate response to external change. There is also a quiet biochemical aspect to honey's stability that becomes more apparent when considering its enzyme content. Enzymes introduced by bees during processing remain active for a period of time, although their activity is limited by the low water environment. These enzymes play a role in transforming nectar into honey by breaking down complex sugars into simpler ones, which increases sweetness and improves stability. Once the honey is fully formed and moisture levels drop, enzyme activity slows significantly, but does not necessarily stop entirely. Instead, it enters a very low activity state where reactions occur at extremely slow rates. Over long periods, especially under fluctuating environmental conditions, These enzymes can still contribute to minor chemical changes within the honey. However, these changes are so gradual that they do not alter its fundamental identity. From a biochemical perspective, honey can be seen as a system where most active processes have been slowed rather than eliminated, allowing stability to coexist with minimal ongoing transformation. If we step back again and consider honey as part of ecological flow, its role becomes even more interconnected with the broader environment. Bees act as both producers of honey and agents of pollination, linking plant reproduction with energy storage. As they move between flowers, they transfer pollen grains that enable genetic exchange among plants. This process supports biodiversity and maintains the health of ecosystems that produce nectar in the first place. Without this ongoing interaction, the availability of nectar would decline, affecting both bee populations and the production of honey itself. In this sense, honey is not just a byproduct of bee activity, but part of a larger feedback loop that connects plant life, insect behavior, and environmental conditions. Each element depends on the others in a continuous cycle, where changes in one part of the system can influence the others over time. Honey sits within this cycle as a stable endpoint of a dynamic process, representing a moment where energy and biological interaction have been temporarily stored in a physical form. If we continue this reflection toward even finer scales, honey also reveals something about how natural systems handle information without central coordination. The transformation from nectar to honey is not directed by a single controlling mechanism, but emerges from many small interactions between bees, enzymes, and environmental factors. Each bee follows relatively simple behavioral rules, yet the collective outcome is highly organized and efficient. Nectar is gathered, processed, and stored in a structured way that supports the survival of the entire colony. This form of organization is often described in systems science as emergent order, where complex patterns arise from simple repeated actions. Honey is the physical result of this emergent process, capturing within its structure the outcome of thousands of coordinated but decentralized decisions. In this way, it represents not only a biological product, but also a demonstration of how complexity can arise naturally without centralized control, simply through repeated interaction over time. And so, when we observe honey in its final form, resting quietly in stillness, it becomes more than just a substance with sweetness or nutritional value. It becomes a convergence point where multiple slow processes meet and remain balanced. Chemistry stabilizes it, biology produces it, ecology sustains it, and physics governs its behavior. None of these layers dominates the others, yet all of them are present simultaneously within its structure. In a calm scientific reflection, honey can be seen as a reminder that some of the most familiar things in nature are not simple objects, but carefully maintained states of balance, shaped by long sequences of gradual change that eventually settle into something stable enough to exist quietly in a single moment. If we continue to follow honey even further into its quiet scientific depth, another subtle aspect appears in the way it interacts with long-term storage environments. Unlike many natural foods that degrade rapidly once removed from their biological source, honey tends to remain chemically stable under a surprisingly wide range of conditions. This stability is not absolute, but it is unusually resilient because of the way its internal structure limits the availability of free water and restricts microbial activity. However, even in this stable state, honey is still influenced by its surroundings in very gradual ways. Temperature fluctuations over long periods can slightly alter the balance between its liquid and crystalline phases. Exposure to air can slowly influence its moisture content. Even light, when present over extended durations, can contribute to extremely slow chemical shifts in color and flavor. These changes do not happen in abrupt steps, but rather unfold as faint transitions that accumulate over time. In this sense, honey does not resist its environment completely. Instead, it integrates environmental influence at a pace so slow that it appears almost unchanged from moment to moment. If we look more closely at the internal structure during these slow transitions, crystallization remains one of the most scientifically interesting behaviors. The process begins when glucose molecules, which are less soluble than fructose, start to come out of solution and organize into small, solid structures. These structures act as initial points around which more glucose molecules can attach, gradually expanding into larger crystalline networks. The speed and pattern of this process depend on multiple factors, including temperature, glucose to fructose ratio, and the presence of microscopic particles that act as nucleation sites. What is particularly interesting is that crystallization does not occur uniformly throughout the entire volume at once. Instead, it progresses unevenly, forming localized regions that slowly expand. This creates a kind of internal landscape within the honey, where liquid and semi-solid regions can coexist during transitional phases. From a physical perspective, this demonstrates how phase changes in complex mixtures are often gradual and spatially distributed rather than sudden and uniform. There is also a deeper thermodynamic idea present in honey's behavior, which relates to the tendency of systems to move towards states of lower energy over time. In its liquid form, honey exists in a relatively stable energy state, but crystallization represents a shift toward a more ordered configuration under certain conditions. This shift does not occur spontaneously without reason, but is driven by changes in temperature or concentration that alter the balance of molecular interactions. Once crystallization begins, it can release small amounts of energy in the form of heat, although this is typically minimal and not easily noticeable. What matters more is the gradual reorganization of structure that accompanies this transition. Even after crystallization has occurred, the system remains stable, simply in a different configuration. This highlights an important principle in physical chemistry. Stability does not always mean lack of change, but rather the presence of a state that persists even after internal rearrangement has taken place. If we step back again into the ecological context, honey also reflects how energy flows through natural systems are temporarily stored and redistributed. In ecosystems, energy is constantly moving from sunlight to plants, from plants to herbivores, and through various intermediate stages of biological consumption and transformation. Honey represents one of the points where this flow is paused and concentrated. By storing energy in a dense, stable form, bees create a reserve that can be accessed when environmental conditions are not favorable for foraging. This buffering capacity helps maintain colony survival across seasonal changes. In colder months or periods of low floral availability, honey becomes a critical resource that supports continuity of life within the hive. From a systems perspective, this is an example of how biological organisms develop strategies to manage variability in resource availability over time, ensuring that energy captured during abundance can be preserved and used during scarcity. If we consider honey at the level of human sensory experience again, its interaction with perception remains remarkably consistent in its gradual unfolding. The sensation of sweetness does not arrive as a sharp signal, but spreads slowly across sensory receptors. The texture prolongs this experience by extending the time during which molecules interact with taste and smell receptors. Even the act of swallowing does not fully end the sensory experience immediately, as residual flavors continue to release aromatic compounds that are detected after the initial contact has passed. This extended sensory timeline creates a perception that is not fragmented into discrete moments, but instead flows continuously for a short period of time. From a neurological perspective, this kind of input allows the brain to integrate multiple sensory signals over a slightly extended window, producing a unified experience that feels smooth rather than abrupt. Honey naturally fits into this pattern because of its physical properties, which slow down both mechanical and chemical interactions during consumption. And when all of these layers are gently considered together, honey continues to appear as a substance defined by continuity rather than sudden change. It is formed through long biological processes, stabilized by chemical balance, shaped by physical laws, and embedded within ecological cycles that extend far beyond a single moment. Even in stillness, it remains part of these ongoing systems, holding within it the slow history of its creation. In a quiet scientific reflection, honey becomes a reminder that many natural forms are not simply static objects, but stable expressions of long processes that have reached a temporary balance, allowing them to exist in calm continuity within the larger flow of nature. If we continue to stay with honey and allow its quiet science to unfold a little further, we can begin to notice how it behaves almost like a bridge between different timescales in nature. On one hand, it is formed through very fast, repeated actions inside the hive. Bees moving from flower to flower, returning, exchanging nectar, and continuously adjusting their behavior based on immediate conditions. On the other hand, the substance that results from this activity is remarkably long-lasting, capable of remaining stable for months, years, or even longer under the right conditions. This contrast between rapid biological activity and slow chemical stability is one of the more subtle features of honey. It shows how processes that happen quickly can give rise to outcomes that unfold slowly, as if time itself is being reorganized into a denser, more extended form. In a calm scientific sense, honey sits at the intersection of short-term biological motion and long-term chemical persistence, holding both in a single continuous structure without needing to separate them. If we look again at the microscopic composition, another quiet detail emerges in the way water is distributed within honey. Even though honey contains relatively little free water overall, the water that is present is not evenly independent but is instead tightly associated with sugar molecules through hydrogen bonding. These interactions create a structured environment where water molecules are not freely moving in the same way they would in pure liquid water. Instead, they are partially constrained, participating in a network of molecular attractions that limit their availability for biological processes like microbial growth. This bound water contributes to honey's stability in a way that is not immediately visible but is fundamental to its behavior. From a molecular perspective, it is not simply the absence of water that matters, but the way remaining water is integrated into the sugar matrix. This subtle distinction helps explain why honey behaves differently from many other sugar solutions with higher water activity, even when their overall composition might seem similar at first glance. There is also a quiet interplay between honey and temperature gradients that becomes more noticeable when considering how it is stored in natural environments. In a hive, temperature is carefully regulated, but it is not perfectly uniform at every point. Slight variations exist depending on location within the structure, proximity to active bees, and airflow patterns. These small differences can influence how honey matures over time, especially in terms of moisture content and crystallization behavior. In warmer regions of the hive, honey may remain more fluid, while in slightly cooler areas, crystallization may begin earlier. Over time, these differences tend to even out, but during transitional periods, they can create subtle variations in texture within the same hive. This spatial sensitivity to temperature reflects a broader principle in physical systems where even small environmental gradients can influence long-term structural outcomes when given enough time to act. If we step outward again into the broader ecological cycle, honey also reflects a kind of seasonal memory embedded in biological systems. The availability of nectar is not constant throughout the year, but follows the flowering cycles of plants, which in turn depend on climate, rainfall, and temperature patterns. Bees respond to these cycles by adjusting their foraging activity and storage behavior. During periods of abundance, honey is produced and stored more rapidly, while during periods of scarcity, consumption becomes more important than production. Over time, this leads to a natural rhythm within the hive that aligns with environmental cycles. Honey as a stored product becomes a record of these cycles, capturing within its composition the conditions under which it was formed. Each batch reflects a specific moment in ecological time, shaped by the particular combination of plants and environmental conditions present during that period. In this way, honey carries within it a subtle form of seasonal imprinting, linking it directly to the broader rhythms of nature. If we return once more to its physical behavior, honey also demonstrates an interesting relationship with pressure and flow. When subjected to force, it does not respond like a simple liquid that disperses quickly, but instead resists motion in a controlled, gradual way. This resistance is due to internal friction created by dense molecular interactions. As pressure increases, honey begins to flow, but it does so in a slow, continuous manner rather than a sudden release. This behavior is often described in rheological terms, which study how materials deform and flow under stress. Honey's rheology places it in a category of highly viscous fluids, where flow is governed by both internal structure and external force. Yet from a simple observational perspective, what stands out is the way it yields gradually, as if every small change in pressure is absorbed and distributed throughout its structure before any visible movement occurs. This creates a sense of delayed motion, where response is always slightly behind cause, giving honey its characteristic slow-moving quality. And when all of these observations are held together gently, honey continues to appear as a substance defined by continuity, balance, and long, gradual transformation. It is shaped by fast biological activity, stabilized through slow chemical interactions, and influenced by environmental cycles that extend far beyond the moment of its formation. Even in stillness, it remains connected to these ongoing processes, carrying within it the layered effects of time at multiple scales. In a calm scientific reflection, honey becomes less like a simple material and more like a quiet convergence of natural systems where movement and stability are not opposites, but parts of the same continuous unfolding that gradually settles into a form we can observe and hold in a single, peaceful moment. As we remain with honey a little longer, allowing its quiet nature to reveal itself at its own pace, we begin to see that one of its most remarkable qualities is not simply what it contains, but how gently it changes across time. In many natural systems, change is easy to notice. Leaves fall, rivers shift, clouds gather and disappear. But honey belongs to a slower category of transformation, one where the passage of time leaves only delicate traces. A jar of honey sitting undisturbed may look almost identical from one month to the next, and yet countless molecular interactions continue beneath that appearance of stillness. Sugars slowly rearrange themselves, aromatic compounds shift in tiny amounts, and the balance between liquid and crystalline structures subtly adjusts according to temperature. These changes are so gradual that they rarely call attention to themselves. Instead, they remind us that stability in nature is often a matter of pace. Some things change rapidly and visibly, while others change so slowly that they seem permanent, even though they are quietly evolving all the same. This slow pace becomes even more fascinating when we think about the relationship between honey and the bees that create it. A bee lives for only a relatively short period of time, especially during the busy seasons when nectar is abundant. Individual bees come and go, generations replace one another, and yet the collective process of honey production continues with extraordinary consistency. The hive persists even as its members change. Knowledge is not stored in the mind of a single insect, but in patterns of behavior shaped by evolution and repeated across countless generations. Each bee performs small tasks guided by instinct and environmental cues, and from these countless modest actions emerges something stable enough to endure long after any individual bee is gone. Honey, therefore, can be viewed as a product not only of biology, but of continuity itself. It represents how lasting structures can arise from many short-lived contributions woven together through repetition and shared purpose over time. There is a similar continuity present in the plants that provide nectar. Flowering plants have existed on Earth for more than 100 million years, gradually evolving intricate relationships with pollinating insects. The colors of flowers, their scents, the shapes of their petals, and even the timing of their blooming seasons have been influenced by these interactions. Bees evolved alongside them, becoming increasingly specialized at locating nectar and transferring pollen. Honey is one of the quiet outcomes of this long evolutionary partnership. Every drop reflects countless generations of adaptation, where plants and insects slowly adjusted to one another over immense stretches of time. Yet despite this immense history, the process itself remains surprisingly simple. A flower offers nectar, a bee gathers it, the hive transforms and stores it, the cycle repeats. There is no urgency in this sequence, no dramatic turning point, only steady repetition unfolding season after season. If we think about this from the perspective of energy, honey also represents something beautifully efficient. The sunlight that reaches a field of flowers is transformed by photosynthesis into chemical energy stored in nectar. Bees then concentrate this energy into a form that occupies less space, resists spoilage, and remains available long after the flowers themselves have faded. In essence, honey is a carefully preserved reserve of energy captured from sunlight and stabilized through biological and chemical processes. It is difficult to imagine a more elegant example of energy storage in nature. There are no machines involved, no external systems of preservation, only the quiet cooperation of plants, insects, and physical laws operating exactly as they have for millions of years. This efficiency is not hurried or optimized in the modern sense. Instead, it is the result of gradual refinement through evolution, where countless small improvements accumulated over time until the process became remarkably reliable. And perhaps that is one of the gentlest scientific ideas hidden within honey. So much of nature operates not through sudden breakthroughs, but through patient accumulation. Tiny changes repeated over immense periods can create structures of astonishing complexity and stability. The hive is built cell by cell. Nectar is gathered drop by drop. Water evaporates molecule by molecule. Honey thickens gradually until it reaches the familiar form we know so well. Nothing is rushed, and yet everything is accomplished. The final result feels effortless, precisely because so many small processes have quietly worked together over time, as honey rests in stillness, holding within it the memory of flowers, sunlight, seasons, and the tireless motion of bees. It offers a quiet lesson about the natural world. Stability is rarely the absence of movement. More often, it is movement that has found balance. It is change proceeding so gently and consistently that it becomes harmony. And in that soft harmony preserved within something as simple and familiar as honey, the long rhythms of nature continue, calm and unhurried, carrying their quiet story forward through time. And if we remain with that idea a little longer, allowing the story of honey to continue at its own measured pace, we begin to notice how unusual it is that something so familiar can contain so many layers of quiet complexity. In everyday life, honey is often seen only briefly, perhaps stirred into warm tea, spread lightly on bread, or resting unnoticed on a shelf. Yet behind that ordinary presence lies an intricate network of natural processes that stretches outward in every direction. The sweetness on the tongue originates in sunlight that fell onto leaves months before. The aroma carries traces of flowers that bloomed for only a short season. The texture depends on the arrangement of molecules moving at speeds too small to perceive. And the honey itself exists because thousands of bees, each following simple biological rules, contributed to a collective effort that no single bee could accomplish alone. What seems simple at first becomes increasingly connected the longer we observe it, revealing that nature often hides its greatest complexity beneath appearances of calm simplicity. This quiet complexity also reminds us that natural systems are rarely isolated. Honey does not exist independently of flowers, and flowers do not exist independently of soil, water, sunlight, and climate. Bees depend on ecosystems that support plant diversity, while ecosystems themselves are shaped by countless interactions among living organisms and their environment. A small change in rainfall patterns can influence flowering seasons. Changes in flowering seasons affect nectar availability. Nectar availability influences the activity of bees, and bee activity ultimately shapes the character and quantity of honey produced. These connections are not dramatic when viewed individually, but together they form a vast network of relationships extending across landscapes and seasons. Honey becomes one visible expression of this network, a small but tangible result of ecological balance maintained through countless interactions unfolding quietly across time. Even at the level of chemistry, this interconnectedness remains visible. The sugars within honey are not isolated substances but part of larger cycles of carbon movement throughout the biosphere. Carbon atoms that once existed as carbon dioxide in the atmosphere are captured by plants through photosynthesis, transformed into sugars, concentrated into nectar, and eventually stored in honey. Those same atoms may later become part of a bee's metabolism, enter another organism, or return to the atmosphere through natural processes. Nothing remains fixed forever. Matter continuously changes form, moving through different stages while preserving the underlying continuity of natural cycles. Honey simply represents one moment within that movement, where carbon, water, and energy have paused briefly in a stable arrangement before continuing onward through other pathways in the future. There is something quietly reassuring in this continuity. Honey does not attempt to resist the larger processes of nature. Instead, it exists comfortably within them, shaped by cycles that have repeated across immense stretches of time. Flowers bloom and fade. Bees gather and store. Seasons change and return. Chemical bonds form and break. Through all of these transitions, honey remains part of the ongoing rhythm, rather than standing apart from it. Its stability comes not from isolation, but from participating fully in systems that are themselves stable because of their ability to adapt and renew continuously. This balance between persistence and change appears throughout nature, from ecosystems to climate cycles to the molecular behavior of living organisms. Honey offers a small and accessible example of this broader principle, allowing us to observe it in a form that is both ordinary and quietly extraordinary. As the hours pass and our attention continues to move gently through these ideas, honey begins to feel less like an object and more like a meeting point between different scales of existence. Microscopic interactions shape its texture. Biological cooperation shapes its creation. Ecological cycles shape its availability. Planetary energy from the sun shapes the plants that produce nectar. All of these layers overlap within a single golden substance that can rest motionless for long periods while carrying within it the history of countless movements and transformations. There is no need for dramatic conclusions when considering something like this. The science itself unfolds softly, one idea leading naturally into another, each revealing how deeply interconnected even the simplest things can be. And so honey remains here, quietly holding the memory of sunlight, flowers, bees, and time itself. Its sweetness is only one small part of its story. Beneath that sweetness lies chemistry balancing water and sugar, biology coordinating thousands of living beings, and ecology weaving together seasons across landscapes. All of these processes continue, slowly and steadily, whether we notice them or not. And perhaps there is something comforting in that quiet continuity, in knowing that long after a flower has faded or a season has passed, a small trace of that moment can remain preserved, resting peacefully in the stillness of honey. And in that stillness, there is yet another gentle scientific idea waiting to be explored. Honey, despite being one of the oldest foods known to humanity, continues to be studied today with the same quiet curiosity that surrounds many natural phenomena. Researchers examine its chemical composition, measure its physical properties, and investigate the remarkable ways in which it varies from one region to another. What they often discover is not a single defining characteristic, but a delicate combination of many small features working together. This is one reason honey has remained so fascinating across centuries. It is familiar enough to feel ordinary, yet complex enough that each closer observation reveals another layer of understanding. The more carefully it is examined, the more it reminds us that simplicity and complexity are not opposites in nature. Very often they exist side by side, hidden within the same quiet object. One particularly interesting area of study involves the subtle differences between honeys produced from different floral sources. Although all honey shares the same general structure of sugars, water, and trace compounds, the precise proportions vary according to the plants that supplied the nectar. Some honeys are lighter in color and milder in aroma, while others are darker, richer, or slightly more complex in flavor. These differences arise from tiny variations in organic compounds that originate in flowers and are preserved throughout the transformation from nectar to honey. Scientists can sometimes analyze these compounds to identify the botanical origins of a particular honey, tracing it back to specific plant communities and regions. In this sense, honey acts almost like a natural signature of place, quietly recording aspects of the environment in which it was created. The landscape leaves an imprint within it, subtle but measurable, carried forward long after the blooming season has ended. If we think about this slowly, it becomes remarkable that a single spoonful of honey can contain information about climate, plant diversity, soil conditions, and seasonal rhythms. The minerals dissolved in groundwater, the timing of rainfall, the abundance of flowering plants, and the activity of pollinating insects all contribute in their own small ways to the final composition. None of these influences dominate the others completely. Instead, they overlap gently, creating a complex but balanced whole. Honey does not erase the details of its origin. It preserves them, quietly carrying traces of the natural world in a form that remains stable enough to be examined long after its creation. This ability to retain information through chemistry is one of the subtle ways nature records its own history without writing anything down. At the same time, honey reminds us that stability is not the same as permanence. Even the most stable natural substances remain part of larger cycles of change. Over long periods, honey slowly responds to its environment. Crystals may form and dissolve, aromas may soften, colors may deepen slightly. Yet throughout these gradual transformations, its essential identity remains intact. This balance between change and continuity is one of the most recurring patterns found throughout science. Stars evolve over billions of years while preserving the laws that govern them. Forests change from season to season while maintaining the structure of ecosystems. Living organisms grow, adapt, and age while remaining connected to the same biological principles throughout their lives. Honey follows this same quiet pattern on its own scale, demonstrating that lasting things are not necessarily unchanging things. And perhaps that is one of the gentlest lessons hidden within its science. Nature does not always seek permanence in the sense of remaining exactly the same forever. More often, it seeks stability through gradual adaptation, allowing small changes to occur while preserving the overall balance of the system. Honey exemplifies this beautifully. It can shift in texture, vary in composition, and respond to its environment, yet still remain recognizably itself through all of these changes. It is flexible without becoming unstable, enduring without becoming rigid. As our attention drifts softly through these ideas, honey continues to rest quietly at the center of them all. A simple substance made from nectar and thyme, a store of sunlight gathered by flowers and refined by bees, a balance of chemistry, biology, and physics held together in calm continuity. It asks for very little attention, yet rewards patient observation with layer after layer of understanding. And perhaps that is why honey feels so timeless. Not because it stands outside of nature's rhythms, but because it reflects them so completely. Slow transformations, quiet cooperation, gentle balance, and the steady unfolding of countless small processes that together create something enduring, familiar, and wonderfully serene. And as the quiet story of honey continues, it becomes easier to appreciate how many of nature's greatest achievements are built not through speed, but through patience. A single bee gathers only a tiny amount of nectar in one journey, often less than a drop that a human eye could easily notice. Yet through thousands of journeys, repeated day after day across an entire colony, enough nectar is collected to sustain the hive and create reserves that may last through changing seasons. The mathematics of this process is gentle but remarkable. Small actions, when repeated steadily over long periods, can produce outcomes far larger than any individual effort might suggest. This principle appears throughout biology, from the growth of forests to the formation of coral reefs, and honey offers a beautifully accessible example of it. Each tiny contribution is almost insignificant on its own, yet together they become part of something enduring and complete. Nature often works this way, not through sudden leaps, but through countless quiet repetitions that gradually accumulate into stability. There is something equally fascinating in the architecture that surrounds honey inside the hive. The wax cells where honey is stored are arranged in the familiar hexagonal pattern that has long captured the attention of mathematicians and scientists. The hexagon is an efficient shape, capable of filling space without leaving gaps while using a relatively small amount of material. This efficiency allows bees to create strong storage structures that maximize capacity while minimizing the wax required for construction. The geometry is not chosen consciously by individual bees, yet it emerges reliably through their collective behavior. Over generations, evolutionary processes have shaped instincts and behaviors that naturally produce these elegant structures. The result is a storage system where geometry, biology, and material efficiency align in quiet harmony. Honey resting inside these hexagonal chambers becomes part of a design that is both practical and mathematically beautiful, a reminder that simple rules can lead to surprisingly refined outcomes. If we pause for a moment on the idea of wax itself, another layer of quiet science emerges. Beeswax is produced by specialized glands and secreted in tiny scales that worker bees collect and shape into the structures of the hive. Like honey, beeswax is a product of biological transformation, created through the conversion of energy gathered from nectar and stored sugars. Its physical properties make it flexible when warm and stable when cooled, allowing bees to mold it precisely into repeating forms. Over time, the hive becomes a living structure, constantly maintained, repaired, and adapted according to the needs of the colony. Honey does not simply exist inside this structure, it depends on it. The wax protects it, organizes it, and creates the conditions necessary for long-term storage. Together, wax and honey form a partnership of materials shaped entirely through natural processes, each supporting the other in quiet cooperation. Beyond the hive, the existence of honey also reminds us how deeply interconnected living systems truly are. The flowering plants that provide nectar depend on sunlight, water, minerals, and healthy soil. Bees depend on those flowers for nourishment. Entire ecosystems depend on pollination to support plant reproduction and biodiversity. Honey sits quietly within this web of relationships, a stable result of interactions that extend far beyond its own boundaries. In a sense, every drop of honey contains a small reflection of the environment that produced it. It carries traces of weather patterns, seasonal cycles, and ecological balance, all preserved within its chemistry and structure. This interconnectedness is one of the most consistent themes throughout science. Very few things exist alone. Most are shaped by networks of relationships that stretch outward in ways both visible and hidden, and perhaps this is why honey continues to inspire such quiet fascination. It is easy to overlook because it appears so familiar, yet familiarity does not diminish complexity. The longer we observe it, the more connections emerge. The sweetness leads to sugars. Sugars lead to photosynthesis. Photosynthesis leads to sunlight and plant life. Plant life leads to ecosystems, pollination, climate, and evolution. Each idea flows naturally into another, without abrupt transitions, creating a continuous chain of understanding that extends far beyond the surface of the substance itself. So honey remains here, resting patiently at the center of these reflections, neither demanding attention nor resisting it. It is simply the result of many small processes carried out with quiet consistency across seasons and generations. Within its golden stillness lies geometry and chemistry, biology and ecology, energy and time, and as we continue to consider it gently, it becomes clear that honey is not only a product of nature, but also a gentle invitation to observe how patiently and beautifully nature builds the world around us, one small, steady change at a time. And if we allow our attention to remain with honey a while longer, another quiet truth begins to settle into view. Much of what makes honey remarkable cannot be understood by looking at a single moment of its existence. Its story stretches across many different timescales at once. The flight of a bee lasts only minutes. A flower may bloom for a few days. A season unfolds over months. A colony may persist for years. And the evolutionary relationship between bees and flowering plants extends across millions of years. Honey sits gently at the meeting point of all of these rhythms. It is made in the present, shaped by the recent past, and connected to a biological history so vast that it is difficult to imagine in ordinary terms. Yet all of that history becomes quietly condensed into something small enough to rest on a spoon, carrying within it a long sequence of events that unfolded slowly across both time and space. This idea of layered timescales appears often in science. A mountain rises over millions of years, while the wind shaping its surface changes from day to day. A forest grows over centuries, while leaves open each spring in a matter of days. Honey follows this same pattern of overlapping rhythms. The chemical bonds within its sugars are ancient in origin, formed through processes that have existed since the earliest forms of photosynthesis evolved on Earth. The flowers that provide nectar belong to plant lineages shaped through countless generations of adaptation. The bees themselves carry inherited behaviors refined across immense periods of evolutionary time. Yet the actual act of producing honey happens quietly in the present moment, through repeated small actions performed day after day. This coexistence of deep history and immediate activity gives honey a special place among natural substances. It is both ancient and new at the same time. If we move gently into the world of perception once more, it is interesting to consider how humans have responded to honey throughout history. Across many cultures and distant places, people discovered it independently and recognized its unusual properties. They noticed its sweetness, its ability to remain stable, and its connection to flowering landscapes. Long before the chemistry of sugars was understood, long before enzymes or molecular structures could be observed, people recognized that honey behaved differently from most other foods. Science has since explained many of those observations, revealing the physical and chemical reasons behind its stability and sweetness. Yet the sense of quiet wonder remains. In fact, understanding often deepens appreciation rather than diminishing it. Knowing how honey is formed does not make it less remarkable. Instead, it reveals the immense number of processes that must align for such a simple and familiar substance to exist at all. There is something else that honey quietly teaches about natural efficiency. In the hive, very little is wasted. Nectar is gathered carefully. Moisture is removed precisely. Wax is used economically, and stored honey serves as a reliable reserve of energy. The entire system operates through countless interactions that have been refined gradually by evolution. Efficiency here is not imposed from above or designed according to a plan. It emerges naturally because organisms that used resources effectively were more likely to survive and reproduce over time. Honey, therefore, is not only a sweet substance or a stored source of energy, it is also a reflection of how living systems adapt toward balance, conserving what is valuable, and organizing complexity into forms that remain stable across generations. If we think about that slowly, it becomes clear that honey represents a particular kind of wisdom found throughout nature. Not wisdom in the human sense of conscious thought, but wisdom expressed through patterns that persist because they work well. The structure of the hive, the behavior of bees, the chemistry of honey, and the timing of flowering seasons all fit together with remarkable harmony. None of these elements exists in isolation. Each supports the others, forming a network of relationships that remains resilient precisely because it is interconnected. Honey becomes a quiet expression of this interconnectedness, preserving within itself the outcomes of many different systems working together over time. And as our journey through these ideas continues softly onward, honey remains unchanged in one important way. It never hurries. Its creation is gradual. Its transformations are slow. Its stability comes from balance rather than force. Even the science surrounding it unfolds gently, one idea leading naturally into the next. Perhaps this is why honey feels so comforting to study at night, when the mind begins to slow and attention turns toward quieter things. It offers an example of how complexity can exist without chaos, how change can happen without urgency, and how countless small processes can work together so smoothly that their final result appears calm, simple, and enduring. And so honey rests, quietly carrying sunlight gathered by flowers, the labor of bees, the patience of seasons, and the steady rhythm of natural laws. Within its golden stillness lies a world of science unfolding softly, inviting us not to rush toward conclusions, but simply to observe, to understand, and to appreciate how gently nature creates some of its most enduring wonders. And as we continue to stay with honey a little longer, there is a quiet realization that begins to surface about how it behaves not only as a substance, but as a kind of natural convergence point where many physical principles meet without conflict. In most systems we study in science, different forces often compete or push against one another in visible ways. Temperature rises and falls, pressure increases and decreases, Motion accelerates and slows, but in honey, these influences appear to reach a kind of long-term agreement. Its viscosity responds gently to temperature, its stability adjusts gradually to moisture, and its internal structure shifts slowly without sudden disruption. Nothing dominates everything else. Instead, everything seems to coexist within a carefully maintained range. This is not because honey resists change, but because change within it is distributed across many small interactions that balance each other over time. In this way, honey becomes a quiet example of how complex systems can settle into stable patterns, not by eliminating variation, but by absorbing it evenly. If we move a little deeper into the microscopic world again, we find that honey's stability is closely tied to the way its molecules interact through weak but numerous connections. Hydrogen bonds between sugar molecules and water, although individually fragile, become collectively significant when present in vast numbers. These interactions form a dense network that influences how honey flows, how it holds shape, and how it responds to external conditions. No single bond defines its behavior. Instead, it is the cumulative effect of countless small interactions that gives honey its characteristic consistency. This idea appears throughout nature. Strength often does not come from a single dominant force, but from many small forces acting together in coordination. In honey, this principle becomes easy to observe because the system is slow enough for its effects to feel continuous rather than abrupt. There is also a subtle relationship between honey and entropy, the concept in physics that describes the tendency of systems to move toward more probable and evenly distributed states. At first glance, honey might seem like a system that resists this tendency, since it remains ordered and concentrated for long periods. But in reality, it exists in a state that already represents a kind of equilibrium. Its high sugar concentration, low water activity, and stable molecular arrangement mean that it is already in a low-energy configuration relative to its surroundings. This is why it does not easily break down or disperse under normal conditions. However, when conditions change, such as when moisture is introduced or temperature shifts significantly, honey begins to move slowly toward new equilibria. These transitions are not sudden collapses of order, but gradual reorganizations that reflect the underlying tendency of all systems to adjust toward balance in response to environmental change. If we step away from physical principles for a moment and return to the biological world, honey also reflects something about how life manages uncertainty. Bees operate in environments that are not perfectly predictable. Weather shifts, flowers bloom irregularly, and resources fluctuate over time. To survive in such conditions, the hive must maintain reserves that buffer against scarcity. Honey serves this function. It is a stored form of energy that allows the colony to continue functioning even when external conditions are not favorable. This strategy is not unique to bees, but they demonstrate it in a particularly clear and efficient way. Many living systems store energy or resources to manage variability, but honey is one of the most direct examples of this principle, made visible in a single substance. It is a form of preparation embedded in biology, ensuring continuity across uncertain conditions. If we continue to reflect on this slowly, honey also reveals something about how natural systems distribute labor without centralized control. In human-designed systems, coordination often relies on planning, instruction, and hierarchical structure. In a hive, however, coordination emerges from simple local interactions. Each bee responds to immediate cues such as scent, temperature, or movement. There is no single controlling entity directing the entire process, yet the collective outcome is highly organized. Honey production, therefore, is not the result of a top-down system, but of a distributed network of interactions that align naturally over time. This form of organization is often more resilient than centralized systems because it does not depend on a single point of control. Instead, stability emerges from the repetition of simple rules followed consistently by many individuals. And when all of this is considered together, honey continues to appear as something quietly profound in its simplicity. It is a substance that holds within it layers of physics, chemistry, biology, and ecology, all operating in harmony without conflict. It is stable, yet responsive. simple in appearance, yet complex in origin, familiar in everyday life, yet deeply connected to processes that span vast scales of time and space. Even as we observe it more closely, it does not lose its calmness. Instead, it seems to reflect that calmness back, as if the very structure of its existence encourages a slower way of thinking. So honey remains here, resting in quiet equilibrium, shaped by countless interactions that continue to unfold beyond immediate perception. And in that stillness, it gently reminds us that nature often builds its most enduring forms, not through force or urgency, but through patient repetition, subtle balance, and the quiet accumulation of small changes that together create something remarkably steady and complete. And as we continue to stay with honey in this slow unfolding of science, there is another gentle idea that begins to appear, one that connects it to the broader behavior of complex systems across nature. Honey does not exist in isolation from change, but it responds to change in a way that is distributed and softened across its structure. In many physical systems, a sudden input of energy or a rapid environmental shift produces an equally rapid response. Water splashes, metals expand quickly, gases disperse instantly. But honey responds differently. Its internal density and molecular structure act like a buffer, spreading any change across many microscopic interactions. This means that instead of reacting sharply, it adjusts gradually, as if every part of it contributes a small portion to the overall response. In this sense, honey behaves less like a single unified object and more like a coordinated field of interactions, where no single point defines the behavior of the whole. If we look more closely at how this buffering effect works, we find that it is closely related to the way energy moves through viscous substances. In honey, energy does not travel freely or quickly. Instead, it is absorbed and redistributed by the dense network of sugar molecules. When force is applied, such as stirring or pouring, that energy is gradually converted into internal motion, heat, and slow deformation. Nothing is lost, but everything is delayed and spread out. This delay creates the characteristic slow movement that honey is known for. It is not resistance in a rigid sense, but controlled dissipation, where energy is gently absorbed rather than immediately expressed. This property is common in many complex fluids, but honey makes it especially visible because of its thickness and stability. It allows us to observe how energy behaves when movement is not immediate, but stretched across time. There is also a subtle relationship between honey and structural order at the microscopic level that becomes more interesting when considered over long periods. Even when honey appears uniform, it contains small variations in molecular arrangement. These variations are not flaws, but natural outcomes of its formation process. Each batch of honey is shaped by slightly different environmental conditions, leading to small differences in composition and internal structure. Over time, these differences can influence how crystals form, how viscosity changes, and how the substance responds to temperature. Yet despite this variability, honey remains recognizably the same material. This balance between variation and consistency is one of the quiet principles found throughout nature. Systems can remain stable even when their internal details are never exactly identical, as long as the overall relationships between components remain within a certain range. If we move outward again to the ecological scale, honey also reflects the way energy and matter circulate through living environments in slow, continuous loops. Plants absorb sunlight and convert it into chemical energy. Insects move that energy between organisms through feeding and pollination. Decomposers eventually break down organic material, returning nutrients to the soil. Within this cycle, honey represents a temporary storage point where energy is held in a stable and concentrated form before re-entering biological processes. It does not interrupt the cycle, but gently participates in it by preserving energy across time. This ability to store and release energy at different moments helps stabilize ecological systems, especially in environments where resources fluctuate with seasons. Honey becomes part of a broader rhythm of accumulation and release that maintains balance across living systems. If we return once more to the human experience of honey, there is also a quiet psychological aspect to its perception that aligns with its physical properties. Because honey is slow to move, slow to dissolve, and slow to change, it naturally encourages a slower sensory experience. The act of observing it, touching it, or tasting it does not produce sudden shifts in perception, but gradual transitions. This allows attention to settle rather than react quickly. In a broader sense, it demonstrates how physical properties of substances can influence the tempo of perception itself. The mind responds not only to meaning, but also to the speed at which sensory information arrives. Honey, by nature of its structure, tends to arrive gently in awareness, without abruptness or fragmentation. And so, as this reflection continues to unfold, honey remains a quiet example of how stability and change can exist together without contradiction. It is shaped by movement, but appears still. It stores energy, but releases it slowly. It reflects its environment, but maintains its own structure. In all of these ways, it demonstrates how natural systems often achieve balance not by eliminating variation, but by integrating it into a stable framework that persists across time. And in that persistence, honey becomes more than a substance. It becomes a small, steady illustration of how nature organizes complexity into forms that remain calm, continuous, and quietly enduring. And as we continue to stay with honey, there is still another quiet layer of understanding that slowly emerges when we consider how it exists across both chemical precision and natural variability at the same time. In many scientific systems, precision and variability often feel like opposites. Precision suggests fixed ratios, predictable behavior, and consistent outcomes, while variability suggests change, fluctuation, and environmental influence. Honey, however, exists in a space where both are present together without contradiction. Its basic chemical framework remains consistent, dominated by glucose, fructose, and a small amount of water. Yet within that framework there is always variation introduced by floral sources, climate conditions, and seasonal shifts. This means that every sample of honey is both familiar and unique at once. The structure remains recognizable, while the details subtly shift depending on where and when it was formed. In a calm scientific sense, this reflects how many natural systems maintain stability not by eliminating variation, but by absorbing it into a flexible structure that allows for controlled differences without losing overall identity. If we look more closely at how this balance is maintained at the molecular level, it becomes clear that honey's internal structure is not rigid but continuously adaptive. The sugar molecules are in constant motion, but their movement is constrained by density and interaction with surrounding molecules. Water molecules are not freely independent but are instead distributed within this dense network in a way that limits large-scale rearrangement. This creates a system where motion exists but is heavily moderated. Over time, this moderation allows honey to remain stable even while small internal adjustments continue to occur. Temperature changes, for example, do not immediately reorganize the entire structure. Instead, they influence local regions first, and only gradually affect the broader system. This layered response to environmental change is one of the reasons honey appears so steady. It does not react as a single unified block, but as a collection of many small interactions adjusting together over time. There is also an interesting connection between honey and the concept of metastability in physical systems. A metastable system is one that appears stable under normal conditions, but can shift into a different state when sufficiently disturbed. Honey, particularly in its liquid form, can exhibit this kind of behavior when conditions change slowly over time. For instance, crystallization may not begin immediately even when conditions are favorable, because the system requires a small initial disturbance or nucleation point to start the transition. Once that process begins, however, it can gradually spread throughout the substance. This delayed response highlights how stability in nature is often not absolute, but conditional. A system can remain unchanged for long periods, while still containing the potential for transformation under the right circumstances. In honey, this potential remains quietly present, even when nothing appears to be happening on the surface. If we return again to the ecological dimension, honey also reflects how energy storage in nature is rarely static, but always linked to cycles of availability and scarcity. In environments where resources fluctuate, Organisms often develop strategies to store energy during periods of abundance so that it can be used later when conditions are less favorable. Honey is one of the clearest examples of this principle in action. It captures energy during times when flowers are abundant and converts it into a stable form that can persist through seasons when nectar is unavailable. This stored energy is not isolated from the environment, but remains part of a larger cycle. Eventually, it is consumed, metabolized, and returned to biological systems in other forms. In this way, honey represents a pause in energy flow rather than an endpoint, holding potential that will eventually re-enter the broader system of life. If we slow down even further and consider honey in terms of perception over time, there is a subtle but important idea about continuity of experience. Because honey changes slowly in texture, flow, and interaction, it naturally extends the duration of sensory engagement. This means that the experience of honey is not confined to a single instant, but unfolds across a short period of continuous perception. The mind does not receive it as a sharp signal, but as a sequence of gradual transitions. This kind of sensory continuity can create a perception of calmness, not because the stimulus is absent, but because it is distributed evenly across time. In this sense, honey does not simply exist in the physical world, it also shapes the temporal structure of how it is experienced, gently stretching perception into a slower rhythm. And so, as we continue to reflect on honey in this quiet and gradual way, it remains a substance defined not by a single property, but by the harmony of many small and continuous processes. Chemistry, physics, biology, and ecology all contribute to its existence, yet none of them alone fully describes it. It is stable without being static, complex without being chaotic, and familiar without being simple. Even in stillness, it carries within it the memory of motion, and even in simplicity, it holds layers of natural interaction that extend far beyond what is immediately visible. In this calm scientific reflection, honey continues to stand as a gentle reminder that nature often builds its most enduring forms, not through abrupt change, but through the slow, steady alignment of countless small processes working together over time. And as we continue to stay with honey, there is still another quiet layer of understanding that unfolds when we consider how it behaves at the boundary between order and randomness. In nature, very few systems are perfectly ordered or perfectly random. Instead, most exist somewhere in between, where patterns are consistent enough to be stable, yet flexible enough to adapt. Honey occupies this middle space in a particularly clear way. Its overall structure is highly consistent, defined by a stable ratio of sugars and a predictable physical behavior under normal conditions. Yet within that structure, there is always microscopic variation. Molecules move randomly due to thermal energy. Small differences in composition exist from one region to another, and subtle environmental influences shape its internal arrangement. This combination of predictable macro behavior and microscopic variability is what allows honey to remain both stable and responsive. It does not rely on perfect uniformity, but instead on the ability to maintain balance despite constant small fluctuations occurring at every level. If we look more closely at the microscopic motion within honey, we find that even in its thick and slow-moving form, molecular activity never fully stops. Temperature, even at room conditions, provides enough energy for molecules to continue vibrating, rotating, and slowly shifting position over time. However, because honey is so dense, these movements are highly constrained. Each molecule is surrounded by many others, limiting the distance it can travel before interacting again. This creates a system where motion exists, but is heavily restricted in scale and speed. Over long periods, this leads to extremely slow diffusion processes, where components gradually redistribute themselves without any visible flow. From a physical perspective, this demonstrates how time and structure interact. The same fundamental molecular motion that occurs in all liquids becomes stretched and slowed by density, producing a completely different macroscopic experience. There is also a subtle relationship between honey and thermal energy that becomes more noticeable when considering how it transitions between different states of flow. As temperature increases, the added energy allows molecules to move more freely, reducing internal resistance and making honey more fluid. As temperature decreases, molecular motion slows and internal interactions become more dominant, increasing viscosity. This relationship is smooth and continuous rather than abrupt. There is no single point at which honey suddenly changes behavior. Instead, its properties shift gradually across a range of conditions. This smooth response is characteristic of many complex fluids, where structure and motion are deeply intertwined. In honey, this interplay creates a substance that feels almost adaptive, adjusting its flow behavior in response to environmental conditions in a slow and predictable way. If we step back into the biological origins again, the role of enzymes in honey formation adds another layer of quiet complexity. Enzymes introduced by bees during nectar processing are biological catalysts that accelerate specific chemical reactions, such as the breakdown of sucrose into glucose and fructose. This transformation not only changes the sweetness profile, but also contributes to the stability of the final product. Because monosaccharides like glucose and fructose are more chemically stable in concentrated solutions than more complex sugars, the resulting honey becomes less prone to rapid degradation. Once the water content is reduced sufficiently, enzyme activity itself slows dramatically. But the chemical changes they initiated remain embedded in the final structure. In this way, enzymes act as temporary facilitators of transformation, shaping the final state of honey before becoming largely inactive within it. This highlights a broader principle in biology, where many processes are driven by temporary activity that leaves behind long-lasting structural outcomes. If we consider honey in the context of environmental variation, it also serves as a reminder of how natural systems respond to changing conditions over time without losing coherence. Climate, rainfall, temperature patterns, and plant distribution all influence the availability and composition of nectar sources. Bees adapt to these changes by adjusting foraging behavior, timing, and resource allocation. The honey that results is therefore always slightly different, depending on the environmental conditions present during its formation. Yet, despite this variability, honey remains recognizable across regions and seasons. This stability within variability is one of the key features of resilient natural systems. It shows that consistency does not require sameness, but rather the ability to maintain underlying structure while allowing surface-level differences to emerge naturally. And as this reflection continues, honey remains a quiet example of how complexity can be organized into a form that feels simple without actually being simple. It is a product of chemical reactions, biological processes, physical constraints, and ecological interactions, all working together across different scales of time and space. Yet when observed in daily life, it appears calm, still, and familiar. This contrast between underlying complexity and surface simplicity is one of the most common patterns in nature, yet it often goes unnoticed. Honey makes it visible in a gentle way, offering a small but complete example of how systems can remain stable, not by eliminating complexity, but by organizing it so thoroughly that it becomes nearly invisible to casual observation.